Good evening, students. Welcome to my PP class. Today I will discuss another important disease, that is downy mildew. Before the discussion, again I want to share something about the coronavirus. You see, coronavirus considered all of you or everybody like made of RNA and DNA. They doesn't have any time while making the contact with the host cell. They doesn't have time to think whether it is coming from a old person or a younger fellow or a boy or a girl or a Hindu or a Muslims or a Sikh or Christians. Least bother. It just entry entered in your system, damage your machinery, and showing the symptoms. So don't confused, and also don't confused with the various type of messages you are getting in Facebook or in WhatsApp, that the if you are drinking the cow urine you will be cured, or you pray for the god longer period then you will be cured, or you there is a temperature is so high then you will be cured, or use saline water and gargling it will be cured. These are all bogus. Only thing is the keep yourself isolated from the others, restrict yourself inside the room, follow strictly the government rules, and of course follow your parent instruction. That is important, and that is the only way we can combat with this crisis. Please and please keep yourself at your house inside the room, do your proper study, complete the assignment whatever given by the teachers to you, and whenever you feel any problem discuss with your teachers, with your friends, of course with me at any time. Okay, so now I will discuss with the destructive disease, that is downy mildew, and before I will discuss downy mildew as a whole, thereafter I will discuss in detail the grape downy mildew. You know, the downy mildew means the downy growth. Downy growth means cottony growth. I already told you in earlier semester that is this downy word is associated with the small tiny hairs found in the cheeks. In small chicks, you will see there is a small hairy growth in on their neck. That growth is called downy growth, and from that these downy words came. So it's a growth, you can see it's a cottony growth. So downy mildew disease, it's a very destructive disease. You will find throughout the world, especially you will find the areas where temperature is minimum. Of course, in tropical and in temperate countries, in our Southern Asian countries, in winter season, the temperature is favorable for this disease development. It's a destructive disease causing almost every crops, including cereals, including your fruit crops, vegetables, flowers, everywhere you will find there is a downy mildew disease. So if you see its significance, then you see that it's a highly destructive disease. It's a highly destructive disease, and uh, it is found maximum in the area where the temperature above 10 degrees centigrade. A warm, moist, and humid environment is required. And this type of environment in India, especially during the September, October, it is quite natural, this sort of climates. So crop loss may be 100% by the disease, and we have some certain data that in US during 1996, approximately 6.25 billion US dollar they used for controlling this downy mildew disease, and where 16.7% were only related with the downy mildew. And the largest percentage of downy mildew control was for the grape downy mildew, because grape is the main crops of Europe and Australia, in America, in your chilies, in New Zealand. There are many places where the grapes are growing for their purpose of liquor, then brine, brandy, etc. And followed by Pseudoperonospora cubensis, it is downy mildew of cucurbits, causing near about 10% share. Okay. In 2004, in USA, there was an epidemic on cucumber, which result near about economic loss $16 million. You can easily understand what is the gravity of the disease. Okay. If you go for the history, you know that the downy mildew disease, it is a tremendous history, because the Professor Miller did, he developed the Bodex mixture, first invented the Bodex mixture against this disease. So uh, in late 1800s, it completely destroyed the grape cultivars in Europe. In recent times, downy mildew of sorghum appeared in the United States. So another downy mildew of tobacco, that is very important disease of Florida. Near about 40 to 90% of damage of young plants you can see by this disease. So severity of losses depend on the prolonged wet conditions, cool weather. If this type of conditions prevails, there is a chances of the disease. So there are many genus which is causing the downy mildew disease in various crop. Bremia lectucea, Peronospora, Haloperonospora, Peronosclerospora, Plasma para, Pseudoperonospora, Sclerosthora, and Sclerospora. These are the main genus causing downy mildew disease on various crop. And all this fungus, it belongs to the oomycetes group. I already told you that oomycetes fungus, it's a pseudofungi, belongs to the Chromista or Stamenophylla, and it is low temperature loving fungus. See the disease in cucurbit, you see there is a downy mildew symptom. In watermelon, you can see the prominent downy mildew symptoms over here. In corn, you see there is a downy growth, yellowing growth. See the condition of the onion plants due to the downy mildew disease. In downy mildew pea, you will see these yellowing, these are related with the downy mildew. In rose also you will find the downy mildew symptoms. In sorghum, you see the downy mildew growth. Sometimes you will find there is a green ear disease that is related with the downy mildew. In soybean, you see there is a downy growth, yellow patches on the upper surface, and you will see the cottony growth on the lower surface. So it infects various crop. So the pathogen, the genus of downy mildew fungus, coming under the oomycetes. I already told you that. And these are the various genus. And the basic criteria to differentiate this genus is the sporangiophore it developed and the branching pattern of the sporangiophore. You see the branching pattern is different. Here is lateral branching pattern. Here is bremia, is different type of patterns. So here in Peronospora dichotomous, in Pseudoperonospora you will find there is a different type. In Sclerophylla you see the verticillate type of the branching pattern. You can see here in a good view. You can see microscopic photographs. You see sporangiophore of sporangiophore of uh, Peronospora parasitica. You see the Peronospora dichotomously branched, but it is from the tip. This is microscopic graph. This is the Peronospora destructive. Here you will see dichotomously like this, then again this, then again this, again this, and there is a formation of this sporangia, which is more or less round in shape. In case of Pseudoperonospora, especially in case of cucurbits, you see it is also, why it is pseudo? You see it is also same thing, dichotomously branched, but the tip are very pointed. You see there the tip is very pointed and only one single sporangia for attached with this. This is not in case of the Peronospora. So that is slight difference, and here you will see little bit pear shape the sporangia, here is a round shape. So these are the basic criteria by which we can differentiate the various type of genus of which causing the Downy mildew disease. In Bremia you see there is a special the sporangia attached in a different, here is a certain knob like structure you will find where there is a projections, and in that projection there is a formation of this spores. Like this, here is a closer view of these things. You can easily see the association of the sporangia. Okay. In case of sclerospora, you see there is a verticillate type, there is a long sporangiophore, then all these branching from the tip, and there is a formation of the sporangia. And plasma paraviticola, you see there is a lateral growth like this. Here you see this. This is the classical differentiation between various type of sporangiophore of Downy mildew causing pathogen. Now there is a very important topic, history. Do you know this fellow? Mildew, who invented the Podex mixture and made revolution in throughout the world for controlling the Downy mildew disease. You see, there is the insect, you see. This Phylogera, it's a dangerous insect of grapevine in Europe. The cultivar facing tremendous problem with this insect, Phylogera, looking like an aphid, sucking pest, but it infects the root system, the root zone system, roots, and totally kills the plants. At the same time, in aerial leaf, you will see there the formation of the galls, huge number of galls that they are forming, and this way it destroys the entire vine or grapevine orchard, this trunk. So controlling these Phylogera insect during 1887, 1878, the European government, they procuring certain materials, okay, root stock were introduced to Europe from North America. They procure this root stock for prevention from that dangerous insect, Phylogera. But they doesn't know that root stock contain the oospore of the Downy mildew pathogen. So at the same time they imported the oospore of pathogen, and these oospore, it shows its talent. That means when it got the proper environment, it germinate, developed the sporangia, huge amount of Jewish spores, and invaded the cells, kill the entire twig, leaves, everything of the grape plants, and become the devastating disease in Europe, especially in France, in Netherlands, in Germany. There was a problems. So controlling that downy mildew was become a challenge during that time. So every scientist, they were trying their level best to find out the new chemicals to control this downy mildew in the orchards, because they are entirely depend on that orchards, and all the breweries like beer and brandy or all sort of things, liquors, they are maintaining or from prepare from this grapes only. So one day, what happened? The Professor Miller, he was visiting the nearby farmer field and find that some mixture of copper sulfate and chemicals, some materials they used by the local growers, applied the nearby plants of the roads to discourage the pilferage or theft, you can say. Okay, pilferage or theft in the passerby, that means nearby person. And so, interestingly, these uh, diseases were less among that area. This gave tremendous attention to Professor Millard. He discussed with the farmer that what is the reason, what you applied in that area, that all the plants are become infected, but these roadside areas plants are not infected. What is the reason? Then the farmer told that we used copper sulfates and lime mixtures, crude mixtures directly on the leaf surface to prevent the pilferage. Millard took that idea and he tested several times in laboratory, prepared the cultures and tested again the pathogen, and thereafter developed the Bordeaux mixtures and somehow managed the problems of downy mildew in European countries. Still, this brown Bordeaux mixture is a very good chemical, thoroughly throughout the country they are using, and due to that, a salute of Millarded people given in Bordeaux to express the gratitude of French people. They prepare a statue in the Bordeaux. There is a place near in Bordeaux in France. So that is the history behind this downy mildew disease. This is the insect. So now we come to the disease. In downy mildew disease, you see all green parts of the grapevine are susceptible, but symptoms is mostly, or conspicuous symptoms you will find mostly on the leaves. Okay. First, you will find irregular yellowish translucent spots limited to the vein, limited by the vein, and on the upper surface of the leaves. You see, this is a chlorotic patches. Here is the chlorotic patches. Here is the chlorotic patches. These chlorotic patches is gradually increasing day by day when the temperature are very congenial. And if you see the corresponding lower surface of the leaf, you will find the white cottony growth. Typically, you will see the white cottony growth. This is the actually downy growth of the pathogen. This downy growth can be, you can easily find in the berries, in the twigs, in blossoms, everywhere. You see the downy growth. And finally, this area become necrotic due to the huge procurement of the food material by the pathogen. This place become necrotic, leaf become dry. At the same time, the fruit also, here also you see there is a dried fruits become dry and collapse. So this is the sequence wise symptoms you can find out. Here you can see the same thing. This is the chlorotic patches initially, then you see the cottony growth. See the cottony growth here, cottony growth. Don't confuse with the powdery mildew. Powdery mildew generally found on the upper surface of the leaf, and it is not the hairy growth. It is just a powder. It is not the powder. It is the hairy growth. You see, it's a hairy growth. It is not the powder. That is the difference. Don't confuse. It is the lower surface of the leaf, hairy growth, and chlorotic patches. In powdery mildew, no chlorotic patches. Okay. So if you see the pathogen biology, Plasmopara viticula, it's a fungal-like organism, belongs to the Umycota, you know very well, and it's a downy mildew fungus. It belongs to the peronosporales. We are considering as a Plasmopara. Okay, Plasmopara. Now, this organism cannot be propagated on artificial media. It's a biotrophic pathogen. Okay, it requires only living host. Sexual reproduction occur through the fusion of Enthridia and Oogonia, you know very well. Then it developed the oospores, hard triple layer oospores, sexual spores. But one thing you know, Plasmopara viticula has only recently been shown to be heterothelic. What is the meaning of heterothelic? Opposite is homothelic. Earlier it was homothelic. Homothelic means it's a thallus where it is a cell fertile. Thallus is fertile, means it has enthridium and oogonium, and they are fertile to each other. But recently heterothelism also noticed, because it depends on the other host. Okay, the pathogen, the male and female gametangium forming in different thallus, different mycelium. Then it coming in contact, and sexual reproduction and sexual reproduction occur. So this is the new thing. Now the mycelium diameter is varies from 1 to 60 micrometer. It is sinusoidal, means without any cross septa or cell wall, without any septa. Okay, and developed intercellularly and developed haustoria inside the cells. The same for more or less all sort of downy mildew pathogen. The mycelium produces sporangiophore under the leaves, and that sporangiophore is coming out from the stomatal opening, and each based on the branching pattern we can find out. Here in Plasmopara viticula the branching pattern is lateral. I already shown you earlier. Okay, and it form white to lemon shaped sporangia. Now these sporangia, when temperature is congenial, mitosis taking place, this sporangial divided mitotically, forming so many nuclei. There is a cleavage inside the protoplasm. Little amount of protoplasm mixed with the nuclei and ultimately form zoospores. That zoospores having two types of flagella, anteriorly longer tinsel flagella and posteriorly shorter hipless flagella. These zoospores easily released from the sporangia and swim in free water. So free water is very essential for dissemination of this pathogen. Now zoospore, when coming in contact with the host, it insists, it loses its flagella, then it forming germ tube, and through the stomata it entering into the host system and developing the disease. You see, in Plasmopara viticula, this is sporangiophore. With sporangia, I told you that it's a lateral branching pattern. This is lateral branching pattern. Lemon-shaped sporangia. You see, here is a bigger microscopic view. You see, the sporangial attached over here. Okay, it is coming out from the stomatal opening. These are the stomatal opening. These are the guard cells, okay, and stomatal opening. From that stomatal opening, the sporangiophore coming out, which is laterally branched, okay, laterally branched. These are the sporangia. These sporangia actually, I told you that it is releasing Jew spores. These are the Jew spores having tinsel flagella and your whiplash flagella. These are the tinsel flagella. This is tinsel flagella. This is whiplash flagella, and infect the plants. In sexual reproduction, you see the mycelium, it developed enthedium and ogonium. These are the things. Then the nuclei are passing from the enthedia to ogonia, and the plasmogamy is gametangial contact. That is, only nuclei are passing from the enthedium to ogonium, not the protoplasmic materials. Then when passing the nuclei, it fused with the Wis pair, and finally it developed the 2N zygote, that is called Wis spores. These Wis spores it germinate, developing the sporangium. Again, that sporangium mitosis taking place, and so many Jewish spores are formed, and by this way infection. This is the life cycle pattern, you can say. So Jewish spores coming from the sporangia, 5 to 15 Jewish spores are coming from the sporangia, okay, and these are interlayer portion I will discuss. Wis spore is a hard triple layer sexual spores, act as a primary source of inoculum, can be survive in a infected portion or in a soil for many years. So there is some new findings that the pathogen related protein by the host plants Physia vinifera during the infection of tuberculosis has been investigated over the years. The host releases many pathogen related proteins during the infection by the pathogen. So there is a PR protein 2, PR protein 3, PR protein 4 genes are induced in the grapevine host during pathogen infections, which encode various type of cell wall degrading enzymes, that is beta 1,3 glucanase, chitinase, etc. Actually, the oomycetes fungus, especially Plasmopara, you all of must be aware that it contains cellulose instead of chitin. So if it is a cellulose, then how it is degrade the cell wall? From where it chitinase developed? It is with the interaction with the host, and the host releases the chitinase enzymes, and that enzyme degrades the host cell itself. So that is an interesting phenomena. There are many wild hosts where the pathogen can survive. These are the all wild hosts. You can say all are related with the vistus family. So disease cycle, here you can see the disease cycle more or less same for all Downy mildew genus. Only difference here, you can see there is a branching pattern of is lateral. If it is Peronospora, that is dichotomously branched. If it is Sclerospora, then verticillate type of branching pattern. If it is Bremia, then you will find lots of knobs like structures on the sporangiophore. So that way, and these sporangia they are coming from this sporangiophore. Then there is a cleavage and formation of the Jewish spores having two flagella. These Jewish spore incest, that means lucid flagella, it germinate while coming in contact with the host, and it penetrate inside the host, then forming the hostoria. You see, these are hostoria, these are all hostoria, these are all hostoria, and procuring the food materials and damage the host cells and reduce the photosynthetic activity. And due to that, there is a formation of the chlorotic patches initially, and finally it developed the necrotic lesion. You see, there is necrotic lesion, it become dry, leaf become dry and damaged. In sexual reproduction, you see these mycelium produce antheridium and oogonium, and these oogonium there is a gametangial contact plasmogamy, then there were the karyogamy, then followed by um, meiosis afterward. This pathogen is 2N, so meiosis occur in developing gametangia here actually. Here meiosis occur, okay? Meiosis occur over here. So it form the wheat spores, and these wheat spores, if the temperature is maximum, the wheat spore can directly germinate also and infect the host, or it germinate then develop the sporangium, then sporangium develop the zoospores, and these zoospore again infect the cell. So this way life cycle is completed. So from here also we can generate it where management strategies is required. See, management require at first we kill the source, that means primary source of inoculum. It may be in the infected twig, it may be in berry or infected materials, or it can survive in the soil as an wheat spores. So all sort of possibilities, that means sanitation, everything can apply over here. Another thing, we restrict the contact of zoospore over here. Okay, we restrict the contact by use of chemicals, by spray, fungicides. If you restrict this, kill these zoospores, so this sort of things will not come and our crop will be safe. Okay. So the survival, where the pathogen can survive, it can survive as a wheat spore in field or in dead plant materials, in the form of mycelium in dead plant materials, and secondary spread is through the conidia, sporangia or zoospores. Okay. Favorable condition, as I already told you earlier, that 95 to 100% relative humidity is required, and temperature more than 30 degrees centigrade, slightly warm environment is required, or relative humidity with warm. Okay. So if there is a rainfall 10 millimeters and 24 period, it satisfied the moisture level of the field, then disease will spread fastly, because if the soil is moisture, then whatever weeds are found in the soil, it germinates immediately and it spread the juice. The incubation period is short at 5 days by mean temperature 20 to 25 centigrades. Okay. So disease forecasting in India, we don't have proper disease forecasting system for downy mildew disease. We are using only chemical spray. So, but in European countries, lots of forecasting model present. Okay. So they are using various type of parameters for modeling spraying timing. What are the parameters? They incorporate the parameters, the temperature, rainfall, relative humidity, leaf wetness, and incorporate all sort of these things with the pathogen biology or germination period or the population dynamics of the spores, and they develop a model, forecasting model, and accordingly they advise to their farmers for proper following of that model, and they can save their crop and they can easily restrict any sort of epidemic. So in France, you will find, you go through this website, is through this reference, Fossier et al. In Austria, Danger in Germany, Huber, and these are the reference. can go through this. They developed various type of model for their country. Unfortunately, in India, we don't have such a model for down mildew of grape. Regarding the management, I told you that field sanitation, that is collection and burning of fallen leaves, proper maintenance of the orchards is very essential. The vine should be kept high above ground to allow circulation and proper aeration. It is important because this will prevent worm, or this will minimize the relative humidity. It will prevent to create the microclimate inside the orchard. So pruning April to May, September, and burning of infecting twigs during September, it is very essential. In India, we have couple of varieties like Umber Queen, Cardinal, Champa, Champion. These are the Red Sultana. These are the resistant variety used. I got some informations, but you go through the research papers and journals, include more informations about the resistant variety if available. No problem for that. The disease can be effectively managed by 3 to 5 prophylactic spray with the Bodex mixture. This is a very good chemicals, 1%, or Fostile Al, aluminum compound 0.2%, or metalaxyl plus mancozeb combination 0.2%, or azoxystrobin 0.1%. Spray 0.1% means in 1 liter water 1 ml. 0.2% means in 1 liter of water 2 ml. 1 ml in 1 liter means 1000 ppm. 2 ml in 1000 liter means 2000 ppm. As simple. Spray schedule with Bodex mixture 1% and other chemicals. There is a spray schedule developed in our country that you will follow immediately after the pruning of the vines. When the new flowers are formed after 3 to 4 weeks of pruning, small tender twigs, at that times you can spray. And before the buds are open, that is very, because the pathogen infect the blossoms and flowers. And when bunches and berries are formed, okay, and during the shoot growth. These are the 5 times, critical stage you can say, where we can spray the orchard or the crop with Bodex mixtures and with the combination with the other chemicals. So student, this is all about the downy mildew of grapes. All other downy mildew more or less same. Thank you.